Today we'll deal with neuron structure and action potential. The general structure of a neuronal cell consists of cell body followed by axon and axon terminal. The cell body has cell membrane, dendrites, and also the nucleus and mitochondria. All the proteins which are required for the entire neuronal cell will be produced by the cell body and it will be translocated to the axon and also to the axon terminal. Axon has myelin sheath, and myelin sheath will be either produced by oligodendrocytes or it will be produced by the span cell. And node of Renwer will increase the speed of the action potential which is propagated by the particular neuronal cell. And between the cell body and the axon, we have axon hillock region. This hillock region will summarize the action potential which is received by the dendrites and it will be propagating the specified action potential downstream towards the axon terminal. The axon terminal has axon end bulb or the synaptic end bulb where the neurotransmitters will be packed in the vesicle and will be present. So these are the two major types of neuronal cell. One is multipolar interneuron cell, the other one is motor neuron cell. The interpolar neuron cell will be connected with the other neuronal cell. So one neuronal cell will be connected with the other neuronal cell in case of multipolar interneuronal cells. In case of motor neurons, the neuronal cell will be connected with the muscle cell and most of the times the neuronal cell will propagate the signal so as to induce the muscle contraction. So apart from those two major neuronal cells, you have different types of neurons based on the structure. One is unipolar neuron, the pyramidal neuron, multipolar, bipolar, and Purkinje fiber. So each are specified for different locations. Accordingly, the functions of the neuronal cell will be covered. Myelin sheath. Myelin sheath is actually composed of 40% of water and 60 to 65% of lipid. The overall protein content is around 20%. The myelin sheath of CNS has high concentration of proteolipids when compared to the PNS. The other important thing is the protein named myelin, which is called as protein zeros. It is also present in PNS. It performs a similar function to that of PLPs of CNS. The oligodendrocyte cells, these are one of the most important cells of the CNS. They perform myelination of CNS neurons. It refers to the process of formation of neuron myelin sheath. These cells, unlike span cells, do not remain attached to the myelin sheath. Thus, no neurolemma is formed. So if there is no neurolemma, the degeneration once happens to the neuronal cell, the regeneration mechanism will not be favored. Span cell. These are one of the most important cells of the human body. They are similar to that of oligodendrocytes of the CNS. They perform myelination of neuron. The outermost layer of the myelin sheath is known as neurolemma. The neurolemma of the neuron formed by these cells contain nucleus and cytoplasm of the cell, which attributes to the regenerative property of the neurons of PNS. It is important to note that one span cell myelinates one internodal area between the nodes of Renwer's. So the regeneration capacity of these myelin sheath and also the entire neuronal cell will be favored because of the presence of neurolemma in case of span cells. Action potential. So action potential is one potential which has depolarization, repolarization, hyperpolarization phase when compared to the normal resting state. So the resting state will have a particular potential difference across a particular membrane, especially the neuronal membrane. And most of the times, the inside of the cell will be negative in nature, the outside of the cell will be positive in nature. And inside the cell, K plus ion concentration will be more, and outside the cell, the Na plus concentration will be more, around 150 millimolar in nature. In case of depolarization, the Na plus channel will be opened up because of the initial threshold and stimuli. The stimuli can be internal stimuli or external stimuli. So the depolarization phase will be favored by the opening of Na plus channel. So there will be influx of Na plus ions into the cell. So thereby inside of the cell will automatically become positive in nature. The outside of the cell will become negative in nature. So the influx of Na plus will happen until there will be neutralization of Na plus in both the sides. So once the Na plus influx is seized or the Na plus channel has become inactivated, the repolarization phase will be initiated. During the repolarization phase, K plus channels will be opened. There will be efflux of K plus ions. So the efflux of K plus ion will lead to the regeneration of negative charge inside the cell and the positive charge outside the cell. So the more amount of K plus ion which is going out of the cell lead to hyperpolarization, which means the potential will become more negative when compared to the resting membrane potential. After the hyperpolarization, again the membrane will retain its resting membrane potential. The overall one single phase of depolarization followed by repolarization and hyperpolarization, we call it as action potential. The time period in which the next action potential cannot be generated, we call it as refractory period. And the refractory period is again classified into two types. One is absolute refractory period. The other one is relative refractory period. In absolute refractory period, we can't able to generate the next action potential at all. Whereas in case of relative refractory period, if we give the stimuli when compared to the previous stimuli little more higher strength, automatically the next action potential will be generated in case of the relative refractory period. Thank you, students.